By the winter of 1944, the Netherlands was trapped. The Second World War was grinding through its final brutal months, and the western part of the country found itself entirely cut off from the outside world. The occupying forces systematically squeezed the food supply. Survival became a matter of strict arithmetic, dictated by heavy stacks of ration carts that carefully restricted every ounce of bread, meat, and dairy. This chart shows the catastrophic decline in food availability. A typical human body requires around 2,000 calories a day to maintain basic function. But during this period, official rations collapsed, plummeting straight down to just 500 calories. People were freezing and they were starving. They scoured the streets for anything edible, waiting in long, exhausted lines for cuts of horse meat or boiling tulip bulbs just to put something warm in their stomachs. As the commercial food system dissolved entirely, neighborhoods turned to communal kitchens. Children lined up with bowls and tin cups in the winter cold, waiting for a single watery ladle of soup to keep them alive. But inside, thousands of women walking those very streets, a completely hidden biological process was trying to unfold. They were pregnant, and their bodies were attempting an impossible task, constructing an entirely new human being from scratch, in an environment that offered almost zero energy to do it. We often think of the womb as a perfectly insulated sanctuary, completely shielded from the chaos outside. But a developing fetus is actually highly sensitive to the mother's physiological state. When a mother is starving, she doesn't transmit memories or words to her baby. She transmits stress. The intense physical trauma of famine creates a flood of specific stress hormones, and these chemical messengers push aggressively right across the placental barrier. In biological terms, this signal functions as an environmental forecast. The world outside is harsh, freezing, and practically devoid of food. This communication triggers a process called epigenetics. If your DNA is the hardware instruction manual for your body, epigenetics is the software layer operating right above it, deciding which instructions get turned on or off. This diagram shows how molecular tags attach to the DNA strands. In response to starvation signals, the fetus fixes markers to its genetic code, altering how genes behave. It targets the genes that control metabolism, proactively dialing down its metabolic rate to run as slowly and efficiently as possible. This response is a desperate, highly sophisticated survival strategy. The unborn child actively rewrites its own operating system to endure the famine it expects to be born into. By the spring of 1945, the Netherlands was liberated. The punishing winter broke, the war ended, and food finally returned to the tables of the Dutch people. Fast forward almost 60 years later. The babies born during that bitter winter are now modern-day adults, living their entire lives in a European society where calories are cheap and abundant. But when doctors began studying this specific group of people, they noticed a stark anomaly. Compared to siblings born just a few years earlier or later, the hunger winter cohort suffered from significantly higher rates of obesity, diabetes, and heart disease. This comparison shows the genetic data of a normal sequence alongside of famine survivors. In the survivors, scientists found the exact same molecular tags that the body had placed there during the starvation conditions of 1944. These bodies were tuned to survive a landscape of scarcity, yet they emerged into a world of plenty. Because their metabolisms remained locked into starvation mode, their systems hoarded every available calorie, a trait that became a health liability in a food-rich environment. A severe nutritional crisis that lasted only 9 months ended up dictating the biological trajectory of an entire generation for over half a century. Our DNA functions as a living archive. It actively records the extreme environments, the deep traumas, and the sheer survival of the people who carried us. For decades, biology class has taught us a very straightforward story. Your parents pass down a set of biological instructions, and those instructions serve as an absolute blueprint for your body. We learn that this code dictates your eye color, determines your blood type, and even establishes your likelihood of developing certain diseases later in life. And as this diagram shows, that entire set of instructions is written using a remarkably simple vocabulary of just 4 chemical letters, A, T, C, and G. That story of inheritance is technically accurate, but it leaves a massive gap in how your body actually operates. To see the missing piece, you have to look at a functional contradiction sitting inside almost every single cell in your body. Consider the physical differences between your tissues. A neuron rapidly fires electrical signals to process information, while a skin cell acts as a flat, static barrier against the outside world. A muscle cell's entire job is to physically contract and generate force, whereas a liver cell functions as a chemical processing plant, filtering toxins out of your blood. Here is the problem. This visual illustrates a core biological reality. Despite performing entirely different tasks, that neuron contains the exact same genetic sequence as the skin cell. The same rule applies here. The muscle cell and the liver cell carry identical DNA blueprints. If the underlying code is 100% identical across the board, why doesn't your skin fire electrical signals? Why doesn't your liver flex? Having a biological blueprint clearly cannot explain the vast diversity of functions happening inside you right now. The answer comes down to a distinction between storage and execution. Possessing a specific gene and actually using that gene are two completely separate biological processes. Think of your entire genome as a massive architectural library, containing every instruction required to build and run a human. When a specific cell needs to perform its job, it does not walk into this facility and attempt to access every manual at the same time. The books on these shelves are simply a passive database. For the system to work, an active reader has to decide which specific volumes to pull down and which to leave securely closed. To become a muscle or a neuron, a cell holds open specific instructions. This diagram shows the cell highlighting required genes, so they are actively read. Meanwhile, the remaining instructions on the strand are kept quiet, physically bundled up and restricted from access. The enormous access control network dictating these open and closed states has a name: epigenetics. Epi is a Greek prefix meaning over or above. This is literally the code above your DNA, actively managing how your genetic database is deployed in real time. It is this overlying system, not the raw chemical letters underneath, that actually dictates a cell's identity and solves the paradox of our diverse tissues. Zooming out to the level of your whole body, this reframes how we view genetic inheritance. The idea of DNA as a rigid, unchangeable destiny is inaccurate. Your genome is a highly dynamic system. Access to the information inside your cells is constantly shifting, opening and closing based on what your body needs to survive. This continuous regulation is the exact mechanism that allows a single, base-level code to successfully construct something as complex as a human being. The human body is the work of a massive epigenetic system, directing exactly when, where, and how those 4 chemical letters act. Biology was once taught as a closed system. You are born with a genetic sequence that remains fixed for the duration of your life. This sequence is composed of 4 chemical letters, A, T, C, and G. Every cell in your body carries this exact same string of information, regardless of its function. Yet this uniformity presents a paradox. If the code is identical, we must explain how one cell becomes a branching neuron, while another becomes a contractile muscle fiber. Epigenetics provides the mechanism for this diversity. It dictates how cells interact with the sequence without modifying the letters themselves. Because cells can vary their interaction with this data, the foundational DNA sequence is only one part of the biological operating system. To understand this control, we look to a mechanism called DNA methylation. This process involves the physical attachment of tiny chemical groups to specific locations along the DNA strand. Introductory biology often frames these methyl groups as a binary switch. The claim is that their presence simply turns a gene off. Biological systems are rarely that predictable. The binary switch is a convenient oversimplification. If methylation isn't a simple toggle, its function must be governed by a logic that redefines how a cell prioritizes its own information. The outcome of methylation is determined entirely by molecular context. The effect changes based on the specific genomic region, the type of cell involved, and the current biological state of the organism. We can view this sorting of information through the lens of a library. In certain scenarios, methyl groups act as organizational signs. They flag specific instructions, making them easier for the cell to locate and process. In other contexts, these same tags function as physical barriers. They wall off certain chapters, barring the cell from accessing the data. While the cell routes and restricts this information, the underlying sequence, the book itself, remains in its original state. Cellular identity is defined by this management of access. Methylation reorganizes biological priorities without needing to alter a single letter of the code. The DNA sequence provides the text, but the epigenetic architecture provides the interpretation. Because this architecture is dynamic rather than fixed, it is vulnerable to the world outside the cell. Extreme environmental stressors can physically reorganize the way a cell manages its library. We see the evidence of this reorganization in the survivors of a severe famine that struck the Netherlands in 1944. Decades later, researchers found a lasting signature in the cells of those who were in the womb during the crisis. The starvation had triggered a specific change in how their bodies managed growth-related genes. Trauma leaves a physical mark on our biological architecture. It transforms human cells into a living record of environmental history, passed down long after the crisis has ended. In the winter of late 1944, a blockade over the Western Netherlands triggered a devastating famine. People were starving in the streets, painting their demands for bread onto the brick walls. The official daily ration in the affected region plummeted. An entire day's worth of food collapsed to an average of just 667 calories. Normally, when a society is pushed to the brink of mass starvation, basic infrastructure collapses. Demographic and health data are among the first things lost in the chaos of war. Yet, in the Netherlands, a strange historical anomaly occurred. The hospitals kept running. The health registries and the vast paper-based bureaucracy remained entirely intact despite the famine. This surviving ledger shows exactly what was preserved: precise dates, numerical accounts of food rations, and meticulous hospital birth records. Decades later, researchers realized what they had. By matching these exact dates, they could trace specific individuals who were developing inside the womb at the height of the starvation. The accidental survival of this wartime bureaucracy gave science a perfectly sealed, tragic natural laboratory to study human starvation. Researchers categorized the babies based on timing. Some were exposed to the famine late in pregnancy, others early, and some around the time of conception. That timing is the single critical variable. When a mother experiences severe starvation during pregnancy, dictates the specific physical outcome for the child. The embryo functions as a dynamic construction site, where biological systems are actively being assembled from the ground up. A fetus's vulnerability depends entirely on which specific physical systems are being built on the exact days the calories run out. To see how this works, we have to drop down from the hospital records to the microscopic moment of early cellular life. At the very beginning, the math is rapid. One cell splits into two. Two become four. Four become eight, eventually forming a dense cluster. These divisions are about assigning purpose. This is the exact window when biological identity forms and the core operating programs are established. The embryo lays down blueprints for systems that will become a brain, heart, and liver. This is managed by the epigenome, the instruction manual telling dividing genes how to behave. Undergoing massive changes, it is highly vulnerable to the mother's lack of calories. When resources are cut off during this window, the embryo must adapt. It begins tailoring its internal systems to survive a world where calories are scarce. Scientists wanted to know if this brief microscopic adaptation would persist into adulthood. The famine did, in fact, leave a permanent mark on the children's cellular programming. When scientists looked at the DNA of the babies exposed at conception, they detected a distinct abnormal molecular signature, a biological record of the famine. They then shifted their perspective decades forward in time, looking at these same individuals as fully grown adults. Long after the 1945 blockade ended, those microscopic molecular scars remained active inside their adult bodies. Human development remains tethered to our earliest environment. The external horrors of history can reach across decades, manifesting in the very code of ourselves. By the spring of 1945, the snow had melted, the war had ended, and the Dutch streets filled with cheering crowds. The famine was over. The severe starvation conditions and the strict ration books that dictated survival during the hunger winter were relegated to history. The Netherlands recovered. Years became decades, and the physical rubble was steadily cleared away. And those unborn children, the babies conceived during the worst months of the starvation, safely grew into adults, seemingly leaving the tragedy of their early development entirely in the past. Human intuition tells us that when a population is fed and decades pass, the body heals. We naturally assume that time and physical recovery wipe the biological slate clean. 60 years later, the scientists returned to those specific individuals, who are now older adults, to study their biology. The researchers had a specific suspicion. They hypothesized that the biological slate was not wiped clean at all, and that the famine had left a hidden mark. But proving that hypothesis presented a massive scientific roadblock decades after the event. How do you separate the biological effects of a prenatal famine from 60 years of other confounding variables? An adult's current health is shaped by a lifetime of different diets, their exercise habits, and their own baseline genetics. To prove the famine directly caused a specific biological change today, researchers needed a natural experiment. They needed a way to completely isolate that brief window of early-stage starvation from every other variable in a human life. They found an elegant methodological solution. They compared the famine-exposed adults directly against their own unexposed same-sex siblings, using them as the perfect control group. Brothers and sisters share a significant portion of their foundational genetics. They also grow up in the exact same household environment. But there is one defining difference. Only one sibling was developing in the womb during the precise window of the famine. This sibling comparison mathematically isolated the variable. Any biological difference discovered between them today must be the direct result of that early life famine environment, and absolutely nothing else. With the variables perfectly isolated, the investigation shifted from tracking broad human populations all the way down to the microscopic cellular level. They zeroed in on a specific target, a region associated with a gene called IGF-2. This is a gene that plays an important role in regulating human growth and development. The results from the lab were striking. The exposed individuals had a detectable molecular difference when compared directly to their own unexposed brothers or sisters. This bar chart visualizes that exact finding. It shows a modest but distinct drop in DNA methylation at the IGF-2 gene for the exposed siblings, compared to their unexposed counterparts. Scientists had successfully detected the physical residue of World War II inside the cells of a modern adult. We need to pause here and clarify a major biological misconception about what this discovery actually is. The famine did not rewrite their DNA sequence. The letters of their genetic code were not scrambled. The trauma did not forge a brand new mutation, and scientists did not discover a starvation gene. The base genetic code survived the historical crisis completely unaltered. If the core DNA sequence didn't mutate, this proves the existence of a secondary, highly reactive biological system operating directly on top of our genetics. That secondary system is DNA methylation. As you can see in this animation, it works through small chemical tags that attach to the exterior of the DNA strand, reacting to the surrounding environment during early development. The historical famine was a severe but temporary condition, yet the epigenetic mark it left behind was incredibly persistent. Ultimately, this tells us that our physical environments do not rewrite our underlying genetic code. The sequence remains intact. Instead, the environment dictates how that code is expressed. Severe early life conditions leave a permanent, measurable molecular footprint that dictates how our biology operates for the rest of our lives. Imagine for a moment the perspective of a newly developing organism. You exist in total darkness, completely sealed off from the outside world. In this enclosed space, you have no eyes or ears. You know nothing about geopolitics, global supply chains, or failing agricultural economies. Yet, you still have to prepare for the environment waiting outside. Your only way of gathering intelligence about that world is through the raw chemistry of biology. Information arrives as molecules. Nutrients, hormones, and metabolic signals constantly flow across the placenta, carrying chemical updates from the mother's body to your own. The womb is not a passive incubator, but a highly sensitive listening post. The developing body uses this chemical data to build a system intended for the world it expects to find. Suppose the signals coming across that barrier indicate a crisis. The chemical updates suggest a severe lack of calories, pointing to an environment experiencing intense starvation. The developing body responds by shifting its own internal chemistry. It prioritizes vital organs and alters metabolic pathways to conserve whatever limited energy it receives. Scientists call this developmental plasticity. It is the biological capacity to alter physical development based directly on environmental conditions. This mechanism is an adaptation for survival. By adjusting its metabolism early, the organism is making a biological prediction about the future. It is hardwiring its cells and tissues to endure the calorie-deprived reality it expects to face on day one. This predictive system carries a vulnerability. We have to ask what happens when the environment changes and the body's prediction turns out to be wrong. Consider a body optimized in the womb to survive starvation. Then, almost overnight, it is born into a sudden flood of abundant, high-calorie food. The cellular machinery programmed to hoard every calorie now finds itself in an environment of plenty. The metabolism optimized for scarcity physically struggles to function in this new context. This collision is known as a mismatch. Recognizing this friction helps explain why certain lifelong health and metabolic trajectories emerge. The adaptations that allowed the infant to survive its initial environment remain locked in place, potentially becoming a physiological liability decades later. Human biology is far too complex to reduce lifelong health to the status of a single gene. The real takeaway is that extreme early environments create persistent molecular differences. They don't dictate our destiny, but they do change our starting point. These chemical responses remain detectable in the blood many decades after the original event. The maternal environment essentially writes the first draft of our internal biological history. Our past is a physical, readable record carried within us. Early development leaves a persistent archive of our history within our own cells. Scan social media today, and you will find a list of extraordinary claims. Creators suggest your thoughts can rewrite your DNA, that your grandmother's sadness lives inside your chromosomes, or that meditation can activate a perfect hidden genome. Science does not categorize these ideas as facts. Epigenetics is real, but it is not magic. Trauma is not archived in your genes like a memory, and your DNA does not change its sequence because of a positive thought. However, the opposite extreme, the one found in older biology textbooks, is also incomplete. For decades, students were taught that DNA was a completely isolated script, a locked set of instructions that remained indifferent to the world outside the cell. The reality exists in the space between internet pseudoscience and rigid determinism. If the environment can influence our biology without rewriting the genetic code, we have to look at the physical structures that sit on top of it. This interaction is managed by regulatory molecules, chemical tags attached to the exterior of the DNA double helix, influencing genes without changing the code. These layers respond to life's conditions. Factors like age, cell type, and environment cause these tags to shift, multiply, or detach. This system is characterized by its instability. Some marks remain stable for decades, while others change by the hour. Furthermore, simply identifying a molecular difference does not automatically reveal its function within the body. This is biology's method for interacting with the world. It allows a cell to listen to its surroundings and adjust its behavior based on what it perceives. These physical, molecular interactions are the reason life experiences can leave marks on a body that persist long after the event has ended. The Dutch hunger winter of 1944 provides a historical baseline for how severe stress impacts this system. During a period of Nazi-imposed starvation, the biological environment for thousands of people changed overnight. In the decades following the famine, researchers looked at the children born during this period. The famous IGF-2 study found that these individuals possessed lasting epigenetic differences compared to their siblings who were not exposed to the famine. While this is often cited as evidence of inherited trauma, there is a specific nuance to the data. The people in this study were physically inside the womb when the famine occurred. This is an example of direct prenatal exposure. The developing cells were living in the same biological environment as the mother, experiencing the nutritional collapse and hormonal stress as it happened. The question for modern biology is what happens in the next generation. If a person experiences a traumatic event years before they ever have a child, can that information still find a way through?
For true inheritance to occur, environmental information would have to physically reach and alter the sperm or egg cells, and then remain intact until the moment of conception. There is a significant biological obstacle to this kind of inheritance. When a new life begins, the body performs an event known as the Great Reset. Biology is designed to reprogram and erase the vast majority of epigenetic markers from the previous generation. This process strips away the chemical tags to provide the new embryo with a clean developmental start. Researchers are trying to determine if any molecular information from a parent's life can survive this comprehensive erasure. The debate is no longer about whether our environment changes our epigenome. We know it does. The question is whether the consequences of an experience can cross this generational firewall. A sperm cell has no brain, no consciousness, and no neurological structures. It cannot remember a war, and it cannot remember hunger. Yet, despite this lack of memory, scientists are finding evidence that sperm cells may carry more molecular data than previously thought. Epigenetics is an active, ongoing conversation between your body and your life. But the question of whether the weight of a father's trauma can pass to his child remains one of the most significant open questions in biology.